In this video, we look at structure and properties of lipids. Lipids are biological macromolecules that contain carbon and are widely found in organisms. They mainly contain nonpolar covalent bonds and are hydrophobic in nature. One component of lipids are hydrocarbons, which are nonpolar molecules that contain only carbon and hydrogen. As a result, lipids are insoluble in water. They also contain a major component called a fatty acid, which is a hydrocarbon chain bonded to a carboxyl functional group. Fatty acids form key building blocks of lipids. This image shows the basic structure of fatty acids. Fatty acids are classified into two types. They can either be saturated or unsaturated. Saturated fatty acids contain only single bonds between the carbon atoms in the backbone, whereas unsaturated fatty acids contain one or more double bonds within the carbon backbone of the fatty acid. These double bonds cause kinks in the hydrocarbon chains of the fatty acids, giving them certain physical properties. Examples of highly saturated fats include butter, which is solid at room temperature, because the saturated fatty acids within butter pack more closely and compactly. Saturated fats also contain extremely long hydrocarbon chains, for example, waxes, which are stiff solids at room temperature. Highly unsaturated fats, on the other hand, are usually liquid at room temperature. Panel A in this figure shows butter, which is a saturated lipid. Beeswax is a saturated lipid that contains long hydrocarbon tails. And safflower oil is an example of an unsaturated lipid. The lipids that are found within cells are influenced by this physical property of being insoluble in water more than their shared chemical structures. They are insoluble because of a high proportion of nonpolar covalent bonds that exist between the carbon atoms or carbon atoms and hydrogen atoms. The three most important types of lipids found in cells are fats, also known as triacylglycerols or triglycerides, steroids, and phospholipids. It's important to note here that all fats are lipids, but not all lipids are fats. Fats are composed of three fatty acids linked to a glycerol molecule. When the fatty acids in fats are polyunsaturated, they form liquid triacylglycerols called oils. Fats are formed when a dehydration reaction occurs between a hydroxyl group of a glycerol molecule and the carboxyl group of a fatty acid. This results in the formation of an ester linkage between the glycerol and the fatty acid molecules. The primary role of fats inside cells is to store energy. As we can see in this image, a dehydration reaction between the hydroxyl group on glycerol and the carboxyl group on a fatty acid leads to the formation of an ester linkage between the two. This process repeats for the second and third hydroxyl groups on glycerol as well, forming a triacylglycerol or triglyceride. Steroids also belong to the family of lipids and are distinguished by a bulky four-ring structure. Steroids can be distinguished from each other by the side groups that are present on the different carbons in those hydrophobic rings. An example of a steroid is cholesterol. Cholesterol is an important component of plasma membranes in many organisms. This is a 3D computer model of the basic four-ring structure of cholesterol. Lipids that are commonly found within cell membranes have both a polar hydrophilic region as well as a nonpolar hydrophobic region. This means that these molecules are amphipathic. The head region contains highly polar covalent bonds, whereas the tail region is comprised of two nonpolar fatty acid or isoprene chains. When added to a solution, these lipids form membranes. The water-loving or hydrophilic regions, called the heads, interact with water, and the hydrophobic or water-hating tails do not. This image shows the basic structure of a phospholipid containing two regions. First, the hydrophilic head, which contains a polar or charged group connected to a phosphate, which is then linked to a hydroxyl functional group on a glycerol molecule. The glycerol molecule, in turn, is connected to two fatty acid chains that make up the nonpolar or hydrophobic tail of the phospholipid. These tails can be composed of either saturated or unsaturated fatty acids. This 3D computer model shows the basic structure of a phospholipid. This concludes an overview of lipids. At the end of this short section, I would like you to be able to answer these two basic questions. Thank you.